In this video, we are going to go over the topics that deal with the second biomolecule, which is lipids. First, let's define what is a lipid. Lipids are biomolecules that contain either fatty acids or steroids. The reality is that when it comes to lipid, there's not one single structure that defines all lipids. As we can see, the structure of a fatty acid is a carboxylic acid, while the structure of a steroid is going to be cyclic structures, which can be cycloalkanes, and it can have other functional groups. What lipids have in common is that they are going to be soluble in organic solvents, but not in water. So this suggests that most lipids are nonpolar. The word lipids comes from the Greek word lipos, which means fat. And these biomolecules are important features in cell membranes, fat-soluble vitamins, and steroid hormones. As I mentioned, when it comes to lipids, there are two major class families of lipids, which include fatty acids and steroids. We are going to dedicate the first part talking about fatty acids, and then we're going to proceed to talk about the most important steroids. When it comes to fatty acids, let's understand that there's different ways to draw them. And in the figure that we see in the slides, we have several ways, and we can write the condensed structures for the fatty acids, and then we can proceed to talk about, or to see, I should say, the structure of a skeletal chemical formula for a fatty acid. Fatty acids are going to be unbranched, and they have a carboxylic acid at the end. So that's why they're called fatty acids. Typically, they're 18 to 18 carbons in length, but there's, of course, fatty acids that are longer than that. Now, because fatty acids have a long part of the molecule that has those nonpolar carbon-carbon and carbon-hydrogen bonds, that's why they're insoluble in water. Because even though this area of the molecule has two polar bonds, the nonpolar character is so large that overall the molecule is nonpolar. Furthermore, fatty acids can be categorized as saturated fatty acids if they only have carbon-carbon single bonds, and unsaturated fatty acids if they have one or more carbon-carbon double bonds. Table 15.1 is crucial that you learn the name of the fatty acids, the carbon atoms, the double bonds, and the structures for the molecules that you see in the table. This table has been split into two slides, and we can observe how in the first slide, which is what we have projected right now, this is illustrating the structures for saturated fatty acids that have 12 carbons, 14 carbons, 16 carbons, and 18 carbons. The other half of the table, we can see it illustrated here, which is representing the monounsaturated fatty acids, and we can observe that they have one double bond. And for the polyunsaturated fatty acids, we have more than one double bonds, and we can see here that there's some fatty acids that have two, some fatty acids have three, some fatty acids have more than three. But again, when it comes to these structures, it's important that you know name, number of carbons and double bonds, and the structure. You may choose to learn the condensed or the skeletal. One of the features of unsaturated fatty acids is that they have the double bonds that I just mentioned. In these unsaturated fatty acids, we have what is called the omega number. In food product that you may have at home, you may see in the label that it states that a particular food product has omega-3, omega-6, omega-9. And what does that omega mean? Well, the omega number for a fatty acid, it's simply the position of the first carbon in a carbon-carbon double bond. So for example, here we have DHA, an important fatty acid in brain development in children. So when we look at DHA and we're trying to determine the omega number or omega term for it, what we have to do is number the carbon atoms starting from the end, that is away from the carboxyl group. So the omega term, as we label it, it starts here. So we go 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, and 22. Those are the 22 carbons in the molecule. So when we're trying to determine the omega number for this fatty acid specifically, we see that there is the start of a double bond at carbon 3, at carbon 6, at carbon 9, at carbon 12, at carbon 15, and at carbon 18. So the omega number for this fatty acid is omega 3, 6, 9, 12, 15, and 18. And as you can see, this omega term states all the places within the molecule when we start numbering at the end opposite from the carboxyl group, the position of the carbon-carbon double bond. It is important to note that when it comes to trends, specifically for fatty acids, melting points for saturated versus unsaturated fatty acids are different. Here we have the example specifically of stearic acid versus oleic acid. Both of these fatty acids have 18 carbons in length, and yet when we look at their melting points, they're going to have different values. Stearic acid, which is a saturated fatty acid, has a melting point of 69 degrees Celsius. Oleic acid, which is a monounsaturated fatty acid, has a melting point of 14 degrees Celsius. So why is it that both molecules, even though they're fatty acids, they're both nonpolar, they have a striking difference when it comes to their melting point? Well, that stems from the idea of the way in which dispersion forces are going to be present in these molecules. The reality is that even though dispersion forces is what talks about the intermolecular reactions or attractive forces within these fatty acid molecules, the reality is that in stearic acid, because these molecules are saturated, the surface area that they have exposed actually allows them to stack better. So in order for us to use an analogy for stacking, it's kind of like stacking Pringle potato chips. Saturated fatty acids can do that. Then, when it comes to oleic acid has a lower melting point because it doesn't have as much as a dispersion for present, because what happens is that the double bonds that are present in the molecule are going to give them less stackability. It's not that they cannot stack, it's just that they can stack less. So just to give you an analogy, it would be like trying to stack Lay's potato chips. They still stack, but not as well. So this difference in stackability is what results in the striking differences for the melting points in these structures. In a few moments, we are actually going to see how this also implies the differences when it comes to state of matter. Let's just first establish that when you have unsaturated fatty acids, they tend to be liquid at room temperature, while when it comes to saturated fatty acids, they tend to be solid in room temperature. And all of these observations also stem from these dispersion forces or stackability that these molecules have inherently. Now, when it comes to fatty acids, we know that there are different forms that, or different biomolecules that has them. And if we revise the original image that we have, when it comes to fatty acids, they are subdivided into waxes, triacylglycerols, glycerophospholipids, and lastly, sphingolipids. Now, we are only going to specifically look into a subset of these, but I would like to encourage you to at least understand that all of these biomolecules, these four, have specifically fatty acids in their structures, as you can see in the following cartoon. Let's now move on to talk specifically about triacylglycerols, which are important biomolecules for the human body. So, what is a triacylglycerol? A triacylglycerol, which is also known as a triglyceride, is the way in which we store fat. What happens overall is that these triacylglycerols in its structures can have 1, 2, 3 components of fatty acids. Sometimes they can be the same, as you can see in the molecule of the left. Sometimes they can be different, as you can see in the molecule on the right. More importantly, these triacylglycerols is how we store fat in order for using it later for generating energy if the body requires it. Even though we are not going to be looking at the oxidation reduction reactions that are involved in order to produce energy from these molecules, it is at least important to understand that this is the way in which we store triacylglycerols for energy. These triacylglycerols can undergo different reactions. Now, these reactions are not new. These reactions have been discussed previously in earlier chapters. So what you see in table 15.3 is just a summary of how you have observed these reactions in previous chapters and how it can actually relate to the lipid structures that we have here. So to at least give you an introduction to it, we are going to talk about esterification, hydrogenation, hydrolysis, followed by saponification of these lipids. Let's get started. The first reaction that we are going to see is esterification. So before in esterification, we learned how a carboxylic acid and an alcohol make an ester. This reaction also happens in biological systems. What happens is that glycerol is your alcohol component. Fatty acids are going to be your carboxylic acid components. And in the synthesis of a triglyceride, in other words, a triacylglycerol, we see that we form 1, 2, 3 ester bonds. These triacylglycerols can be further reacted, not in the body. This specifically is done in industry, where they can be hydrogenated in order to create food products like shortening or margarine. Hydrogenation of a triacylglycerol, specifically one in which it is unsaturated, leads to the formation of a saturated fatty acid. If we look closely at the reaction itself, This carbon-carbon double bond, each of the CH groups are going to receive the hydrogen on each of the fatty acid components, and they will turn into CH2s. That's why you see that at first we have 7 plus 7, that's 14 CH2s. When these two molecules turn into an additional 2 CH2s, we have a total of 16 CH2s in the hydrogenated product. It is important to note that this reaction also produces another organic product. The side product of this reaction is called a trans fatty acid. And that comes from the idea that most unsaturated fatty acids are cis, because that's how naturally occurring fatty acids are present in nature. And one of the things that happens during this process in this reaction is that instead of cis, the catalysis of the reaction turns the product from cis to trans. That's why you may observe that when products like margarine are available commercially, in the label, it says no trans fat because that side product has been removed. Next, these triacylglycerols can be hydrolyzed. Now, hydrolysis, that it is performed under acidic conditions or by the catalyst called lipase, results in the formation of a glycerol molecule and 3 fatty acid components. These 3 fatty acids, understand that they are going to be protonated, so we have full carboxylic acids. These molecules then can undergo a reaction that is called beta oxidation. And that beta oxidation in the human body is what creates energy in the form of that important biomolecule that is called ATP. Remember that ATP is something that we are going to see later when we talk about nucleic acids. The other type of hydrolysis that can happen to triacylglycerols is one in which you experience in the laboratory. If you have a triacylglycerol, then you can boil it in base and you obtain glycerol, but instead of the fatty acid in the form of a carboxylic acid, you can form a carboxylate ion. This carboxylate ion is a structure that we better known as soap. So specifically, triacylglycerols, when you do base hydrolysis of the esters that are present in the molecule, this is known as saponification, and it results in the formation of soap. Let's now move on to learn about another member of the fatty acid family or molecule that contain fatty acid, which is known as a phospholipid. When it comes to a phospholipid, let's understand that phospholipids are very important because these are going to be the most abundant lipid that it is present in the cell membrane, which is the barrier that surrounds many of the cells in our body and is what allows things to come into the cell and to leave the cell. When we look at the phospholipids, we can think about them in terms of a cartoon in which there is a glycerol molecule connected to 2 fatty acids. And in addition, that glycerol molecule has a bond to a phosphate followed by an amino alcohol. So when we look closely at this, but in terms of organic chemistry, we are going to observe that here are the fatty acids. You can see this one is saturated. This one is unsaturated. And then the last part of glycerol connects to phosphate, followed by an amino alcohol. In order to draw these large molecules, one of the cartoons that is utilized to represent phospholipids is the following. in which you have a representation for the polar head and you have a representation for the two fatty acid tails. If the polar head is not represented by a rectangle, sometimes it is represented by a circle, still with the two tails. So in order to color code this in something similar, you can envision that blue-green area is the polar head and the two tails are also highlighted in orange. Since these biomolecules are present in the cell membrane, let's actually see what the cell membrane looks like. Cell membranes in animal cells are going to be an important component that it is made of those phospholipids that I just illustrated in the previous slide. These phospholipids, as you can see, form what is called a bilayer, because when we look closely at the structures, the polar heads are facing outside of the cell and inside of the cell, and then the corresponding tails of each phospholipid are facing each other in the middle. Let's understand that the plasma membrane not only has phospholipids, there can also be carbohydrate chains, proteins, which is the biomolecule that we will be looking at next. We can have other lipids like cholesterol. And based on the polarity of the functions, remember that the polar heads, since they're polar, they're known as hydrophilic regions because they can interact with water. The non-polar tails that we can see in the middle, because they're non-polar, they're going to be afraid of water. So that's why that region is known as hydrophobic. In order to explain why is it that the plasma membrane has all these structures in it, we always think about it in terms of what is called the fluid mosaic model of the cell. And this model simply explains the structural components of biological membranes. The reason why this is called fluid is because when it comes to the phospholipids, typically what we observe are unsaturated fatty acids. The reason why this model is known as mosaic is because the variety of the components, and as I just stated, remember that there's sugar, remember that there's cholesterol, remember that there's protein, remember that there's at least 2 different types of lipids in them. One of the main roles for this plasma membrane is allowing the entry and the exit of certain substances inside or to leave the cell. So it is important to understand what are the different types of transport that can happen in cell membranes. There are 3 different types of transport that we are going to study. The first one is diffusion. And when it comes to diffusion, this specifically is going to allow particles from higher concentration to lower concentration to move inside of the cell. Typically, diffusion is going to happen to small molecules that include oxygen gas, CO2 gas, and even water can partake in diffusion. The other mode of transport is going to be facilitated transport. In a facilitated transport, what happens is that we have a protein channel that is going to increase the rate of diffusion. And we can see that right here. Now we have like a tunnel that is going to allow these molecules to come in. Facilitated transport is actually observed when we have monosaccharides like glucose entering the cell. Lastly, we have active transport. In active transport, understand that the molecules are going to move in, okay? And these molecules are specifically going to be ions. They're going to move against a concentration gradient, which is what we observe here. But the key for active transport is going to be the use of energy. So that energy molecule that I keep on talking about, ATP, is going to be utilized in order for active transport to occur. Now that we have talked about fatty acids, let's move on to talk about steroids. When it comes to steroids, steroids are going to have what is called a steroid nucleus. And that steroid nucleus is going to be this network of cyclic molecules that are together. We have a 6-member ring connected to a 6-member ring, connected to a 6-member ring, connected to a 5-member ring. The steroid nucleus must have the connectivity of these ring systems in that pattern. Where do we have steroids? Well, they can be synthesized in the body. And specifically, I want to mention cholesterol, which is the main steroid that is made in the body. It's very important. Cholesterol is obtained from meats, milk, and eggs. So it will be obtained from diet, but it can be synthesized by liver. Why is cholesterol so important? As we observed previously, it is present in cell membranes, but moreover, it can be actually utilized in order to make hormones and even make vitamins like vitamin D. The problem with cholesterol is that if you have it in high levels, it can actually clog arteries. We have a particular biomolecule that it is in charge of transporting cholesterol in the body. In that, we can observe here, that molecule that transports cholesterol is known as a lipoprotein. And a lipoprotein is going to allow that cholesterol to be deposited in certain areas or transported back to the liver in order to deal with it. When it comes to cholesterol, let's at least learn some facts about it. Not all cholesterols are going to be the same. We actually have what is called good cholesterol and what is known as bad cholesterol. Good cholesterol is known as HDL, which is a high density lipoprotein. And the reason why this is known as good cholesterol is because it's going to transport cholesterol to your liver, okay? And it's going to prevent the buildup in the arteries. It actually removes cholesterol from your bloodstream. But what if an individual, instead of having good cholesterol, has bad cholesterol? What that means is that at the molecular level, this individual is going to have a larger number of low density lipoproteins. And why low density lipoproteins is known as bad cholesterol is because it actually deposits cholesterol in arteries. And because it continues to deposit it, then that leads to the formation of plaque. And that could result in a heart attack or even a stroke in certain individuals. So it is important that the human body, in order to correctly process cholesterol, has a larger amount of good cholesterol and a lower amount of bad cholesterol. But both molecules are actually necessary in the body in order for cholesterol to be transported to the specific organs that require it or the different biological systems where it needs to be present.